So now I just want to finish my last lecture because it remains some slides concerning the last pathway. But already yesterday, some part of this pathway had been well presented, so it will be easier for you to understand or perhaps to complete. I have chosen so to show different other examples than examples that have been discussed with students here yesterday. So phenylpropanoid pathway is also a very important one. There is not only one pathway because you have now already well understand that there is a common part and after there is some specific parts. So now I think you are quite confident in this type of organization. So I would like to discuss with you of one that is very important, the pathway leading to lignin, production, because it is one of the most important phenylpropanoids in plant cells. And also after just one or two examples in flavonoids and polyacetate, in which we can find the example also of resveratrol, as we have seen yesterday. Okay, so I start. As you have seen already now that the phenylpropanoid pathway have a common part, that it is initiated by Shake-MAT pathway, so now it's quite known by all of you. And also it's the condensation between some derivatives that come from pyruvate metabolisms, that come from the primary one, and also Shake-MAT pathway, in particular, this molecule, picomaroyl coenzyme A, that it is a very efficient molecule. And with this addition of coenzyme A is very interesting to be able to interact with other type of molecules. So this is a way for this acid to be more efficient during its transformation, by obviously some specific enzyme, in particular this one, chalcone synthase, that is the first enzyme that permits to generate the first precursor of flavonoids pathways, the chalcone. After that, there is lots of different branches that can be initiated depending the type of enzyme that are present in the cells. It's always the same scheme, for leading to the four different type of flavonoids. You have now in mind all the diversity, we have in this type of molecules. So here, two enzymes are very important. The first one that enter in this pathway, la PAL, and the second one, the chalcone synthase. We will see again, future, what can happen when we modify the expression, for example, of this type of molecule, according, the transgenic, way, in order to see if we can modify something in the production of specific metabolic pathway branches. Yes. So as you have now this large scheme of the pathway, what happens with this type of specifically molecule lignin? Lignin is a polymer of several units of monolineol. Monolineol are phenylpropanoids that are not unique. There are different type of monolineol, and it is the assembly of this monolineol that lead to lignin. But the assembly can be completely different from one plant to another one, and even in one plant from one cell to another cells, Because lignin, it is a component very important of the cell wall. It can be accumulated in a large part or in a small part. It depends on the type of tissue, of the cells. And also, this biosynthesis of lignin occurs along all the life of the cells. It can be stimulated, it can be stopped, depend on the situation of the plant and of the cell is at one moment in particular. It's a way to reinforce, the cell wall. Lignin make the wood, so it's quite very strong. So depends of the necessity for the cells to be stronger than another one. So it's a very dynamic metabolisms. It's not just at the beginning and after it's finished. It is always modified. So this monolineal come from always the same precursor, phenylalanine, and as I have shown you just before, we can produce this specific first precursor, p-coumaroyl coenzyme I. And what happens in this particular metabolic pathway? There is two types of enzyme that are able to convert this first precursor in different other precursors that lead to at least 3 main branches of this pathway. One is dedicated to the production of one of the first monolineal, what it is called unit H. I don't want to explain each name because it will be long, okay? And after that, you can see that from the same precursor, we can also produce different two other precursors, which is caféoyl-CoA or ferru-oyl-CoA, and both of them lead also to two other units named G unit or S unit, S for serindil, G for guanylyl, and P for p-hydroxylphenyl. Okay, that's all. And all of these three molecules can be converted, okay, via some peroxidation by peroxidase or lacase. Lacase are more copper oxygenase. Do you remember? I present them the first lecture. And they are very important because they finalize completely the formation of this monolineal and also their assembly to produce some lignin. What I want just to show you here is that this was the view of this metabolic pathway we had at least perhaps 6 or 7 years ago. But as some new enzyme have been discovered between, in fact, they have completely reviewed a part of this metabolic pathway. It's only a way for me to show that the pathway are not completely fixed, because as soon as we discover a new enzyme, it can completely reorganize, okay, the view of we have of a particular metabolic pathway. So the metabolic pathway leading to this type of monolineal today have been completely reviewed, and this is at what moment what we think about this pathway, okay? So always we have exactly the same first part of the pathway leading to the precursor, and what the difference is that this precursor can be taken charge as it was shown before, leading to the three precursors leading to subunit H, C, G, and S, exactly as the same way we have seen. But what was found, I don't know, but I think it's 5 or 6 years ago at maximum, these enzymes that are able to hydroxylate also directly this first precursor and lead, in fact, to different other intermediates that can permit to produce also different other type of unit of monolineal, less important, not by their role, but by their amount in the cell, okay? But by this way, it can explain the complexity of the monolineal families that can be found in cells. And we don't know if perhaps new enzyme can be discovered in order to implement new branch of this metabolic pathway, leading, for example, for new type of unit that could be assimilated to monolineals. So it means that, in fact, we are always thinking about a new discovery in this area to establish how this type of complex metabolic pathway could be modified or expanded. Okay. And what happens in the cells? So it's like you have already now well understand. Not all the step of this metabolic pathway occurs in the same place in the cell. Here it is divided between cytosol, vacuole, and also the cell walls. In fact, the first step of this metabolic pathway starts near the nucleus on the reticulum endoplasmic membrane, because there is a lots of P450 proteins that are membranes proteins. Okay, so they need to be anchored on the reticulum endoplasmic membrane. So lots of the first step occur at this moment. After some of other steps, including hydroxylase, for example, occur directly in the cytosol, leading to the production of monolineal units. But these monolineal units are glycosylated and they are accumulated after in the vacuole. Always the same type of scheme, okay? And in the vacuole, they wait. When the cell needs to produce new lignins, what happens? This monolineal, glycosylated monolineal, are able to transfer from vacuole to the cell wall directly, so they go out the cell, in the cell wall. And here, in the cell wall, it exists lots of also enzymes, so it's not a passive, okay, compartment. There is also lots of enzymatic activity in the cell wall, where these monolineal are deglycosylated, so they became again alone without any protection, and they are taken in charge by lacase and also peroxidase in order to polymerize by themselves and to produce lignins. But they can also give another type of molecule named neolineals, but it is not the same. And all of these cells are able to increase, okay, the lignin contents of their cell wall. But the lignin contents mean that perhaps it's not exactly the same composition at each moment, okay, of the biosynthesis of the lignin. This is why, for example, young cells, they have the cell walls, but they need to expand because they are young. So the lignin is thin, the cell wall is quite thin, and lignin is not so strong, okay, as for mature cells, and particularly so cells that are from vessels, such as xylem, where there is normally a very large cell wall. And in this case, the lignin needs to be more strong. So the composition of the different monolineal units can be also different. So by this way, even with only some small bricks, named monolineal, you may have a different type of arrangement that lead to different characteristics of the cell or the cell wall at what moment in particular. Okay? So here, what it is interesting is that I say to you that these metabolites are produced in the vacuum, in the cytosol after they are accumulated in the vacuole, and after they are I don't want to say nothing. After they have to be transferred in the cell walls, but you need to understand that this transfer are not so easy to do, are not so natural, because there is some membrane that are here to identify vacuole, and this membrane is not so easy to transfer. Okay, so you need to have some specific transporters that help this movement of metabolites from one compartment to another compartment. And when we are interested by deciphering a metabolic pathway, this element is also very important. Not only the enzymes that are able to transform one precursor in one product and so on, but also when we realize the complexity of the architecture of the metabolic pathway only in one cell, it's important to identify the intermediate proteins that are not enzymes, here transporter, that permit, okay, the transfer from one compartment to another one. And there is also a very large set of different type of transporter family in plant cells. And some are very dedicated to specific type of molecule, so are less specific, depend of what type of transporter you have. So it can be also very important to identify them, because they could be also considered as some molecular tools in case of we want to modify different things. For example, you can imagine if we if we increase or if we decrease the number of transporters, for example, here at the vacuole, it could lead to a less or a higher accumulation of these monolineal intermediates in the vacuole. And so this accumulation is also interesting because it means that they can be quickly mobilized in case of necessity to produce some lignins. If you have not this transporter, the mobilization could be less efficient, and in fact, you may have a less strong lignin. That could be a very interesting thing. For example, if you want to have some trees for producing papers, it's quite interesting to produce, but lignin is not so interesting to have in this type of trees because it needs more steps to produce paper. So one idea was, can we decrease lignin production or to have a lignin less stronger than the other in order to have less step to produce some papers? And for example, they have tried to modify the level of monolineal production, but also they have tried to find a solution by limiting this transport from one to the other compartment that have an influence on the final lignin mobilization, production by monolineal mobilization in the cell. So you can see that, in fact, we can act on the regulation of this type of biosynthesis pathway, not only by acting directly on the enzymes, but also by acting by the, the transport and also the different way that this metabolite have to take in order to arrive at the final step to produce here lignins. Okay. So here, just to show you the last type of the synthesis of lignins. So here are the two important enzymes at the end of production of this complex molecule. Here, just to show you what type of modifications they are able to produce, and also how these are the 3 or 4 main units. I'm sorry, because I thought I had put one example of a lignin molecule, which is a complex one, very important one, but you can find them easily on internet if you take a look at that. Here also, it's another illustration of what I have started to explain you, is that when you have 5 different type of plants, pine, alfalfa, Arabidopsis, poplar, cones, and you can see that if it's a color code, you need H in gray, you need G in pink, okay, and you need S in green, and you can see you can have different composition, okay, of lignin, depend of the type of plant. And what it is interesting here, for example, poplar, which is a tree, poplar used for different things, okay, in particular, so wood for different application. They have a big portion of S unit instead of G unit, while some other plants, more herbs, such as Arabidopsis, have more this unit than S unit. So means that this show at the point of what lignin is quite different from one plant to another one, and this was taken as an advantage to identify new genes or new enzymes that are involved in this type of complex metabolic pathways. And several years ago now, it has been shown also that sometimes when monolineal are polymerized not only in lignin but in lignan, which is another type of phenylpropanoid molecules of families. This biosynthesis is catalyzed by several enzymes, but they need to be helped by a new category of protein, what was named dirigent protein. It was quite interesting because it was the first time it was shown that in fact it exists some intermediate proteins that are able to link, for example here, a monolineal, which is one of the brick of building a polymer, and they are able to orient this monolineal in a good way in order to help the enzyme to have a good catalytic process to lead to the lignan formation. So mean that this dirigent protein play a role also in order to orient this specific precursor to increase the success of the polymerization at the end. So means not only enzyme but transporter, dirigent protein, and so on. You can now imagine that around the metabolic pathway, it exists lots of other type of proteins that could play a very important role in this final biosynthesis. So this is why here we focused on metabolic pathway, but in the cell it is completely more complex than only a metabolic pathway. And when you are working on this type of project, you need to take in account quite all of these systems. So even if you are interested by looking at for specific enzymes, you have to integrate that they are not alone in the cell, not alone in the compartment of the cell. So they have some interaction with different other molecules in the cell, so that it is a complex way to integrate. But it's very interesting also to discuss. So this is the example of lignin I wanted to discuss with you because it is one of the domains in which there is a lots of research, and every years, I can't say every month, but every years they discover something new. They explain, they rearrange different things. So it is quite interesting to have for you as an example in mind. But here I have put again, this scheme just to record you different other type of flavonoid classes. And here just to show you, you know that now very well, the different three first steps. And if you want to, not to have a specific phenylpropanoid pathway, but always the complex architecture of from one precursor, how many flavonoids can be do. We can summarize this in at least 3 or 4 specific activities. Structural modification. So, for example, if you have in the first, in the first scaffold, of the first phenylpropanoid, generine, you have 3 rings, A, C, and B. And what happens is, if you have a double bond between the C2 and the C3 here, situated here, you can enter all the pathway of flavon and flavonols. So means that just one of the first modification is an initial step of two specific branch. And after, if you have some dehydroxylation in particular here, for example, means that you enter into what we call the dehydroflavonols. So just one modification is a way to make a dichotomic branch from a start point. And if we continue, and here if we have also between always the same type of two carbon, C and C2 and C3, if we have an isomerization with a specific aryl group, it's not important to know what is it, but you may have all the isoflavonoid pathway that it is initiated. So by this way, you can understand that in fact, it's not so complex to see how these branches are able to be separated. Okay, it depends only of enzyme activity. So means that if in one cells or in one plant, okay, you modify the activity of one of these enzymes, you can completely rearrange the balance between all of the branches. So you can increase a part of this phenylpropanoid while you have a decrease of another part. And means also that the cells is able to adapt according the necessity of the moment, okay, production of this type of phenylpropanoids. It's exactly what was explained at the beginning of the lecture. These molecules are produced in reaction of modification of environment. So they need to be ready to be produced. So this is why, in fact, they are able to produce all the precursors. And at the end, when it is necessary, just one enzyme is necessary to activate it in order to enter in the pathway, in the part of the pathway, okay, leading to the specific, for example, phytoalexin that could have the final action for the plants to survive to the modification of the environmental condition, for example. So the complexity, the apparent complexity of this metabolic pathway is in fact a way for the plants to be ready against every modification and to be ready to have an answer, a quick answer, in order to be less sensitive to the modification, to the environmental modification. So this is why it's quite interesting. And we can study this type of attitude, by not only by the fact to study metabolomic production, okay, so by screening, for example, the metabolites that are produced at what moment, but also we can observe the production of the enzymes, okay, that are able to stimulate one branch, second branch, or third branch, for example, of this pathway. By this way, we can follow the amount of transcripts hemirinia encoding the protein, okay, that are produced at one moment. So it's a way also to have an idea of how this pathway are managed by the cells depending on the type of signals. For example, if you apply the signals such as highlight, UV, or another biotic signals such as a pathogen, virus, fungi, and so on, you can see that it is not exactly the same enzyme that are stimulated. So it's a way to link which type of metabolite correspond to which type of signals in order to have a more physiological idea of what is the role of this type of metabolite or this type of metabolisms in general. Okay, don't worry. We will see that. So here, just to summarize, because I have I don't want to forget two other type of family that are very important, anthocyanins and the proanthocyanins. and here it's always the case. You have these two branches have the same common precursor, and only the fact of the choice of one or another enzyme between here in S and LER lead to one or to another type of metabolites. Not all are produced at the same times. And you have seen with Professor Smita that we can prepare some cell suspension, for example, cell culture. And what can be very interesting is that, in fact, when you have an idea of why in the plant this enzyme, for example, is stimulated instead of this one or the contrary, you can reproduce this type of signal on your cells in order to enhance, for example, production of anthocyanin instead of proanthocyanin. And it is always the case with the others. So it is why it's interesting to have an idea of what happens in the real life of the plant, okay, to have an idea of what type of signal can be tried to apply to a cell suspension, for example, in order to stimulate more specifically a part of the branch instead of all the plant metabolic pathway, okay, that it is not what we are looking for sometimes. So this is why it's interesting to know, okay, what are the organization of this pathway and what are the enzymes that are very interesting. And whatever, if you can't succeed, because sometimes it is the case, even if you have applied lots of stimulation in order to improve a specific part of the pathway, but unfortunately you don't succeed to enhance the production of a specific metabolite, after you can move to another type of modification, transgenic modification, so to make some GMO cells, and you can act directly on this type of enzyme by increasing, overexpress them, or at the contrary, to down the expression of them, or you can silence completely also this type of genes that completely lead to the modification of metabolic contents in the cells. So by this way, you may have lots of possibility to modify the final contents of this metabolite, which is always what we are trying to do to produce specific extract or specific molecule that could be after purified to different application, mainly pharmaceutical one. Well, and also in this case, I just wanted to speak about polyacetate, because in a class of phenylpropanoid, there is some propanoids that come from this special activity, polyacetate synthase. And you can see here that it is what I said to you. Depend of the enzyme that are involved. You have seen yesterday that instead of chalcone synthase, if you have some stilbene synthase, okay, you can produce with the same precursor two different type of final molecules, resveratrol as it was shown, but also as we have seen how all phenylpropanoid. But the precursor here is a polyacetylated precursor. So means that in fact, before it's very interesting to see how this type of intermediate is produced before. And it is one of the most interesting part of the pathway, because it's also a way to increase this type of precursor in the cells in order to improve the production of one or of the other type of molecule. And here also, it exists lots of cell suspension from grape, for example, that have been already established, in which the aim is to produce high amount of resveratrol, because you have seen that this molecule have lots of good application. And not only the fact to have only cells that are able to produce resveratrol, we want to have cells that produce very large concentration of this molecule, larger than what we can be found in normal cells in the plant. And here it is the type of the two way we can use to access to this type of concentration is or to enhance the quantity of precursor, or to stimulate specifically the stilbene synthase in the cell instead of chalcone synthase, which are always present, whatever the type of cells, because phenylpropanoids are important for each cell. While stilbene synthase are present only in cells able to produce resveratrol. So this is why we need to make balance between both in order to enhance this type of molecule. And what was not said yesterday, but I can add some, okay, is that in fact, resveratrol is a very interesting product, but in fact, it was found that polymer of resveratrol, because resveratrol is able to be polymerized twice, 3 or 4 times, so it can to become very complex molecule. And it is also some stilbene synthase that are involved in this type of polymerization. And what was found is that, in fact, like antifungi or antibacterial activity are more efficient with polymerization of resveratrol instead of the resveratrol molecule alone. So now it was also interesting to see if we don't know all the stilbene synthase. We are always looking for genes that encode this type of enzymes. But what it was shown is that this stilbene synthase responsible to production of resveratrol is not able to produce the polymerization of resveratrol. So means that it seems to have also here a family of stilbene synthase genes that will be able to have all the activity of polymerization. We don't know if there is only one that will be able to polymerize in 1, 2, 3, or 4 units, or if it's necessary to have one specific stilbene synthase enzyme for each step of the polymerization. So there is a lot of job to do, okay, for identifying this. And it is very important because if resveratrol is produced in a high amount in a cell, its polymerized forms are very low in the cells. But as this molecule have been found to have very, very important antifungi activity, there is a lot of project that aims to use this type of molecule to produce some natural pesticide. Okay. So by the way is so it will be interesting to produce large amount of this molecule or to enrich, extract with this type of polymerized resveratrol molecule instead to have only resveratrol. So this is why there is a lot of biotechnological project aiming to have this type of production of this molecule. Okay, just to show you that just with this type of short part of the pathway, we can discuss a little bit about the importance of what we can do, what we would like to do, what could be the next and the future research in this type of small part of the pathway, leading the use, okay, not only of this molecule, but all of its derivatives. And just to finish, I don't want to explain all the glucosinolate pathway, because glucosinolate pathway I have already shown you. It was at the beginning of my talk when I have spoken after of the different enzyme families that are involved in the metabolic pathway. I show you a big picture with a lot of row with different color, and I say it's in brassicacea, the glucosinolate metabolic pathway. You have seen that there is now lots of enzymes that have been identified, but it remains a lot of other ones that need to be identified too. But just to show you what is a glucosinolate, okay, a glucosinolate, it's a sulfur heteroside or what we call a thioglucoside. Means that you have here, okay, a genuine, and this one is linked with a glucose via a thiol, okay, link, and it is also linked with a sulfate group, okay, with linked with here an amine. This is the natural structure of a glycosinolate. This type of organic compounds are very important because, in fact, they are quite small and they can be easily hydrolyzed in the cells. That can permit to give new molecule of glucose too. It's a way of glucosylation, deglucosylation, but also they can give the volatile sulfur. This is why it is this type of molecule, when they are hydrolyzed, they gave this not good smell, okay, of different organic matter. Okay, so this is important to know that they play a very important role by accumulating glucose and a possibility to release them also in the cell. And if you don't know perhaps some of them, they are responsible for what we say is bitter, okay, or the pungent flavor of many compound foods, in particular mustard. This is why sometimes when you eat directly mustard, you have in the nose, okay, some specific, it's not so pleasant, okay, the sensation we have. It becomes from this particular glucosinolate. But you can find them in different other type of vegetables that are currently used by us, and in particular in cauliflower. And people don't like this type of molecule in cauliflower because it gave, for babies, for example, it gave not a bitter taste of the vegetable. So this is why they have tried to find a way to decrease the metabolic pathway of glycosinolate in cauliflower in order to have less amount of this type of molecule in the plants. So it's not by transgenic approaches. It is by natural selection, crossover, okay, between different genotype of cauliflower. But now they have some specific biochemical markers in order to predict if the metabolic pathway will be more expressed or less expressed, that could lead to less glucosinolate, for example, in the final fruits, okay, or legumes that you are able to cook after. So there is so many examples where this pathway, even if we don't know all the steps, okay, but we have some knowledge of the pathway that can be served as markers, are followed to decrease, or in other case to increase, depend of the application. Okay, this production in order to have more in agreement with our own gust, in fact. Okay, so this is also, we can do lots of things without making some transgenic plants. Okay, and by knowing this type of metabolisms, we can have more markers to follow, okay, what we are doing by natural cross-linking with natural way to select new varieties of plants food, for example. So another way also that it is interesting to know for you, glucosinolate are very interesting to enrich soils. So, and I don't know if it will be the case here in India, but in Europe, there is specific fields that are where mustard are grown, but they are not harvested for different other applications. They are laid directly on the soil, okay, because by this way, this all the components, and particularly glucosinolate, okay, are incorporated into the soil, that increase the amount of what we call isothiocyanates that are very efficient against soil pests, fungus, and nematode, and different other pathogens that are in the soils. So what we observe now is that there is a lots of type of strategy leading not to make some chemicals, okay, addition in the soils, but between two campaign of normal production of different plants, okay, we have this type of activity to put some specific brassicacées, okay, because they are able to produce lots of glucosinolate. Mustard is one of the most used plants for this type of application, and by this way, it's a natural way, okay, to make a new dynamic in the soils in order to protect the future plants that will be, the future seeds that will be put in the soil. So here are the type of the products that are able to produce from glycosinolate, nitrile, organic thiocyanate, and isothiocyanate, a very small molecule that are very have a long life in the soils, so it's very efficient, not for several months but several years too. So this is not necessary to do that every year, but every 2, 3, or 4 years it will be enough. Okay, and it is very, very, very efficient. Well, and also here mustard that are used for this type of biofumigation. It is the term we use for this type of culture. It's not the same mustard used for foods. In fact, they have been also selected to produce specifically glucosinolates that are able to produce more derivative of this type, and also it was a way the knowledge of the glycosinolate pathway was a way to have some markers to see which variety of mustard will be the most interesting to use for this type of application instead of using a mustard just for food application. Okay. It's just a way for me to show you that knowledge of metabolic pathway are not only interesting to know what the production of one metabolite in particular, but can be also used to apply plants in general for several other type of application too. Okay. Yes, I just want to finish with a part of the metabolic pathway that have been now well deciphered, okay, of the glycosinolate pathway. Here you can see that you have also different type of compartments, cytosol, chloroplast, but you are quite convinced now that it is not simple. You need to be an architect, okay, to build this metabolic pathway. And it is always the same organization. First one, elongation chain, it is the chain elongation machinery from the an amino acid that have been chosen as precursor. Here, the second step here is to build the core of the glycosinolate structure. Okay, so there is some specific enzymes that are already been well known. You see here mainly monooxygenase, and at the beginning and at the end mainly glucosyl transferase, because it is just the step before the last and final part of the pathway. So here there is always some accumulation step of the intermediate of the pathway. And the last one is the secondary modification of the amino acid side chain, leading to the different variety of glycosinolate that exist in the cells. So now I think you are well informed about what is the metabolic pathway, how they are mainly organized. There is not one but a diverse metabolic pathway leading to the so diverse amount of metabolite. And if you have any question, I can